understand them, those are 6 questions you should have had no problem with. So can't say that's true for everybody, but in theory, if you understand the quizzes and the tests, you should do that much better on the subsequent tests. Okay, so next I want to talk about the unknown report, the unknown lab report. That is due, we have to check the schedule. Does anybody have a schedule in front of them? October 30th, right. So the last day of October, so about 3 weeks away, I think 2 weeks away, 2.5 weeks away. I have the instructions. It's not too crazy, nothing that you guys should be able to do fairly easily. All right, so let's just go over the instructions for a couple minutes. And if you guys have any questions, let me know, and then I'll show you the template for writing the report. I've set up a template where basically you just have to fill in the blanks, and it should be fairly simple. So let me actually pull it up here. So here's our CPORT page. It's under course lessons where we keep our PowerPoints. Get on to today's date. There's something called the unknown report template. Just a Word document. Okay, so at the top, enter your name, enter the number of your unknown, and then here in the boxes where you want to write what you think your unknown is, the species name. Okay. And then here are the sections I'll talk about right now. So the first section is the bacterial significance. So if you look on the report instructions, bacterial significance, so you identify your unknown. In this bacterial significance section, you want to write 2 paragraphs. Okay, I've bolded 2 because I want to, and that'll come into the formatting part of your grade. If you basically follow basic instructions, if you write 3 paragraphs or 1 paragraph, I'll take off a couple points. So I want 2 paragraphs. Why this specific bacteria is important. Okay. So examples, any diseases that it can cause. How it's used in biotechnology research. So you do your own outside research to find some important facts about your bacterial strain. Okay. The procedure section, which is procedure and rationale, is the next section. So this is really where you explain how you determined what your unknown was. So you explain what experiments you did in the lab, what the results were, and how that led you to the conclusion of what your unknown is. Okay. So you don't have to write every experiment that we've done in lab. So if a certain experiment that we did in lab wasn't important for you in identifying your unknown, you don't have to write about it. Okay? So we've all done a lot of experiments, but let's say catalase, you didn't need to know if your unknown was catalase positive or negative to determine your unknown. You don't have to write about the catalase experiment. Everybody understand that? Okay. So you only have to write about the ones you needed to determine your unknown. Okay? So again, it should include some rationale. So what were you trying to determine by doing each experiment? So you don't want to say something like, I followed the dichotomous key chart, because that's not really why you did it. But something more like, I wanted to see if my unknown could use glucose and produce gas. Therefore, I did the carbohydrate fermentation lab where I used phenol red broth and inoculated my unknown. So a quick sentence or 2 explanation about why that experiment was done, what you're trying to determine by that experiment, and what the results were. Okay. So there's a little bit more detail there. You can read that. Important here, you should also mention which experiments were repeated to confirm results that were not clear. So again, we have some time next week where you can repeat any experiments that you might have had unclear results for or that you want to repeat. So if you did repeat it, you do want to list it here and why you repeated it. In bold, very important, for each experiment you talk about, you want to say what your positive and negative control was for that experiment. Okay. If you did not use one, so not every experiment do we provide a positive and negative control. Sometimes we just do positive, sometimes we don't do any. If we didn't do any controls for that, I want you to figure out what could have been used as a positive or negative control. Okay. Another important thing, in order to get full credit, I want to see some attempt to use tables, graphs, pictures. Some of you take pictures of your different tubes. If you didn't do that, that's fine. You can put your data in some kind of table format or some kind of graph in order to represent it. So I want some kind of graphical representation of your data. I don't want just words. Okay? So there's a couple of fairly simple ways to do that. Very important, don't include images from websites. So don't say something like, let's say your unknown fermented glucose, so it turned the broth a yellow color, which is the experiment we're doing today. And you didn't take a picture, so you go on Google Images and you find a tube with yellow broth in it and say, this is what might look like. I don't want to see any images from the internet, okay? If you didn't take the picture, just don't include a picture, okay? If you do that, it's falsifying data, and then you'll get a 0 for that section, okay? Next is the BLAST sequence. So you guys haven't been introduced to BLAST yet, but if you look at your schedule for today, there's a take-home, some take-home work to do. I think it's exercise 17 in the lab notebook. It tells you how to use BLAST and how to get a sequence from BLAST. So what I suggest is you do that exercise at some point before you try to write this report, figure out how to use BLAST based on that take-home work, and then use that information in order to get this information from your unknown. Okay? So for the BLAST sequence, you'll basically list a DNA sequence that's specific to your species at 60 to 120 nucleotides long. I want you to provide a sheet. So for BLAST, when you put in this information and you get this information as an output, I want you to print it out to show me where you got your information. Then you'll have, these things will all be found in that printout, the locus, the gene product, and the gene function of that sequence that you picked. Okay, so there's numerous amounts of different sequences for any species. So this could be, that's why I want to see the printout. I want to see where you got the information from. Okay, and it's pretty straightforward. Again, I suggest you do exercise 17 at some point over the next week or so, and then you'll be familiar with BLAST, and then you can do that to figure it out. Okay. There's an example here of what type of information I'm looking for. Okay, verification experiment. Verification experiment will require a little research. So basically, this is where I want you to write about a new experiment that is not mentioned anywhere in the list of experiments that can be run in the lab. Okay, what that means is on your dichotomous key, remember there was that list of experiments that could be done in the lab that you guys could use to make your dichotomous key. So I want you to do some outside research and find an experiment that's not on that list that would be good in determining what your unknown is. Okay? So I want you to write about that experiment, how it's done, what your expected results would be for your unknown, and then importantly, I want you to also write what positive and negative controls you would use for that experiment. Okay? So you could use Burgie's, you could use any kind of online search to figure that out. Okay, whatever you use to figure that out, whatever source you use, you make sure you want to include it in your references. Citations should be included throughout the report, so anytime you get any kind of information from an outside source, you should be citing it and including it in your references section. There's a link here about what kind of format I want. I'm not going to be a big stickler about format, about references, but as long as I can get the information I need. There's many ways you can do it. You can put little numbers along the way as you do your references. You can put the author in parentheses in the text, however you want to do it, as long as I can figure it out. Okay. Important note here in italics in the middle, if you reference any of the procedures in the lab manual, which you will be as part of the results section, you do want to make sure you include the lab manual as a reference. So the presentation is supposed to be between 3 and 4 pages, and you need to address all the sections that are given above, including references. Include a cover sheet, so I don't need anything fancy like one of those folders where you put everything in. Just a cover sheet that has, just your name and date and class and stuff just stapled to the front is fine. Formatting, 11 to 12 point size font, 1-inch margins. I don't want you playing with the margins all a bunch, so you can squeeze more information into 4 pages or stretch out less information into 3 pages. Okay, and then in bold, I put this in bold because there's at least a couple people that forget to do this every year, and it's an easy way to lose points. Make sure you format your bacterial names correctly, so the first time you mention it, you write out the full name in italics, and then at each subsequent time, you just put the first letter and the second name. Okay. The point breakdown, if you correctly identify your unknown, it's 10 points out of the 100. So again, it's not make or break whether you get it right, although it helps if you do. The presentation and format, again, this is following just the basic directions of how I want it laid out, 10 points. Bacterial significance would be 20 points. The procedure section, which will be your biggest section, would be 30 points. The blast sequence, another 10 points. Verification experiment, 10 points. And then the citation, 10 points, for a total of 100 points. Okay, so this report is basically the same weighting in your grade as the exam we just took. So it's a good way, if you didn't do as well as you wanted to at the exam, this would be an easy way to make up some of those points. Okay. So again, this is pretty significant, about 10% of your total grade in the class. So you do want to do the best you can and make sure you follow the directions closely. You might want to reread this a couple times and make sure you're following all the little details, because that's an easy way to lose points. Okay, so again, the template's online. I prefer you just use the template. It makes it easy for me to grade if everybody's using the same template format. Okay, any questions? So I know a couple people just came in who didn't get some of the instructions. No, just text.
How much of a reference do you want us to put into report when we refer procedure from lab manual?
How many references?
Like how details? So the
Just usually, an experiment should be able to be described in a sentence,
okay,
maybe 2.
okay,
I don't need a bunch of background about the experiment. Like I don't need what's in your lab notebook.
you don't need to know the procedure, just like the name.
Just like basic procedure, like in order to figure out if my unknown fermented glucose, I inoculated my unknown in a phenol red broth and incubated for 48 hours to see if there was gas or a color change.
And did we tell our result on right after that as well?
Okay, yeah, so I found my result that there was a gas bubble produced, therefore
Positive.
it was positive for that. Okay, anything else?
Do we have to know our unknown first before, because I tried to use the blast last night, but it's asking me for my unknown.
So you can do it with any strain just as practice. Once you know your unknown,
Okay.
then you can do that with your unknown. So yeah, so for the lab book, for exercise 17, where it teaches you how to use the blast, it tells you to do your unknown, but at this point, you probably don't know what your unknown is, so you can just pick any strain just as practice and do exercise 17 on just whatever strain you want. And then once you know your unknown, you can repeat it with your unknown. Okay, so you guys should have all the information you need to determine what your unknown is by next Wednesday. Okay? So today, besides the experiment we're doing today, which will tell us a lot about your unknown, it'll tell you what type of carbohydrates it ferments, which I think you guys will help you guys figure it out. So besides doing that this afternoon or this evening, I also suggest you get together with the other people that have the same unknown as you, and you guys put together a strategy about what other tests you might want to do or what tests you might need to repeat, because I need to tell the lab coordinator by the end of the night tonight. Okay, so again, I'm going to set aside some time in lab today for you guys to figure that out. So yeah, we're doing that experiment today, the carbohydrate fermentation. We're doing the IMVIC tests next week, which again is like 3 or 4 different tests combined into one section. So you'll get a lot of information over the next 2 weeks that should hopefully guide you in the right direction about your unknown. Okay? Anything else?
What's your last teacher? Last number.
3. Yeah, you're not, I know.
One last thing before we start the lecture. So Wednesday, I was thinking a little bit over the weekend about how to handle Wednesday. And I know originally I told you guys we were going to start class late on Wednesday. What I have thought about, I looked at next week's lectures, and they're fairly short lectures next week. So what I'm going to do is just cancel lecture on Wednesday. Okay, so we won't have any lecture on Wednesday. You guys can just show up at 7 o'clock. So we'll just start with lab. Okay, we'll do no lecture. And I'll take the lecture that was planned for Wednesday, which was biotechnology, and we'll do that plus the 2 lectures we had planned next week will just be covered over both days next week. Okay, so we'll basically have 3 lectures covered over 2 days.
So our quiz will only be over.
The quiz will only be on today's lecture, which actually works out because today's lecture is very long and has a ton of information. This is the, I told you guys a couple weeks ago that there's a couple lectures in this class, 2 or 3 lectures in this class that are like crash course lectures of like complete other subjects. So today is your crash course lecture on molecular biology. Okay, so quiz on Monday will only be on this lecture. It might not be a full 10 questions. It might be like closer to 7 or 8. However many questions I do, I'll have, however many less questions than 10 I have on this lab, there'll be that many more questions on the quiz the following week. Okay, so if I get 8 questions on Monday's quiz, the next quiz will be 12 questions. Okay, it'll even have to be 20 questions on those 2 quizzes. Okay. All right, so again, no lecture Wednesday, just show up for lab. And so you guys have a couple options. You can just show up on Wednesday at 7 and just go for lab, or you could show up at your regular time and it'd be a good, hour and a half, 2 hours for you guys to do any last minute changes to your dichotomous key and get it in working order. I don't need you to turn it into me until lab, so you guys, if you want to show up at regular time and work on it for an hour and a half and finish it up, you can do that. It's up to you guys. However you guys want to use that time, but again, we'll push Wednesday's lecture into next week. Okay. All right. So today we'll be looking at microbial genetics again, which is a lot of basic molecular biology. So if you guys had any molecular biology, you'll be a little bit ahead here. If not, I'll try to take it down to a more basic level. If I'm going too fast or you need me to repeat something, just please stop me in the middle and we'll go ahead and do that. Okay, so this is just a question that we've actually talked about before, but just wanted to pose it at the beginning because we'll kind of explain the phenomenon of how this happens in general. So E. coli is a normal microbiota species that's found naturally in the human large intestine without any negative side effects. And when it's there, it's actually beneficial. However, there is a strain called E. coli 0157H7 that produces a Shiga toxin that actually causes very severe food poisoning. Okay, so how did this specific strain of E. coli acquire this gene that's actually a toxin from a different bacterial species? Okay, so we talked about this before, and we're going to get into all the basics of how this happens. Okay, so start with some definitions. So genetics is just the study of what genes are, how these genes carry information, how that information gets expressed, and how those genes get replicated. A gene is a segment of DNA that codes a functional product, usually a protein. And in our case, we'll solely be looking on genes that make proteins. A chromosome is a structure of DNA that carries the genes. And a genome is basically a combination of all the genetic information in the cell. Genomics is the study of genomes, the molecular study of genomes. A genotype is the specific genes of one organism. So if you're talking the genotype of this species versus the genotype of this species, you're talking about the specific genetic information and how they differ between the two. Then the phenotype is the expression of genes. So for instance, genotype for someone that has blue eyes, they'd have slightly different genes from someone who has brown eyes. And so the genotype would be what is the actual difference in the genetic information from a perspective of the DNA. The phenotype would just be that one person has brown eyes, one person has blue eyes. So it's the physical traits, okay, rather than the genetic traits. Okay, so here's actually a picture of an E. coli showing that's basically been lysed open, and so all the DNA has been spilled out all over the place. So this just shows one single circular chromosome from one single E. coli cell. You can see how much genetic information is there. When it's in the cell, it's obviously packaged much tighter. Again, you can just see exactly how large this chromosome is. So here's something called the genetic map of a chromosome. So again, bacterial chromosomes are all circular, so that's a very important thing to remember. What a genetic map does is it takes that circular chromosome and tells you where all the very important genes are located on that chromosome. Okay. So you can see here, they have them basically grouped together by what those genes do within the cell, what metabolic process they belong to. So all the blue genes are part of amino acid metabolism and so on and so forth. What you can see is a lot of the genes that are part of the same pathways are kind of grouped together on the chromosome, and we'll come back to that at a later point. But again, it's basically a mapping of where the genes are physically located on a chromosome. Okay, so we'll be looking at 3 different ways that genetic information gets used within a cell. Okay, so again, genetic information meaning DNA, a circular chromosome for bacteria. So one thing that DNA can do is it can get expressed into the proteins. So when it does that, the DNA gets converted to RNA in a process called transcription. And the RNA then gets converted into a protein in a process called translation. And we'll go over both of those processes. Okay, so that's one thing that DNA does. Another thing that DNA can do is it can recombine with different cells, with other cells, not reproductively, but in other ways. So within the same generation, DNA can recombine to form new genetic reinformation. That's called recombination. We'll talk about that. And then lastly, DNA can just be replicated in the process of a cell replicating. So a single DNA chromosome can turn into 2 chromosomes during the process of a cell dividing into 2 cells. Okay, so we'll look at all 3 of these: expression, recombination, and replication. So here's the basic structure of DNA. Okay, so it's basically a polymer, which just means a chain of multiple nucleotides. So the base structures are all called nucleotides, and there's 4 nucleotides in DNA. Adenine, thymine, cytosine, and guanine, and they're usually just represented by their first letter, so A, T, C, and G. You can see here, A, T, C, and G. Okay, so DNA is a double helix associated with protein. So the double helix basically means there's 2 strands that are complementary to each other, that bind to each other, and kind of wind like a winding staircase. Okay, and we'll show you what that looks like. And the backbone is made up of a sugar-phosphate combination. The sugar is deoxyribose. So here's the sugar. It's this 5-pentagon structure, and the phosphate is here. So the 2 strands that make up the backbone are made up of these sugar-phosphate combinations, and then these bases, the nucleotides, are in the middle. So you kind of think of it as a ladder, where these would be the outside of the ladder, and then the bases would make up the steps of the ladder. Okay. The 2 strands are held together by the bases, and they're held together by hydrogen bonds. And important, they bind specifically. So an A base can only bind to a T base, and vice versa. And a G base can only bind to a C base, and vice versa. Okay, so A cannot bind to C or G. G cannot bind to A or T, so on and so forth. Okay. And the strands are what's called anti-parallel. So what you can see is they go in opposite directions. So if you see this pentose ring has an oxygen at the top, you can see in one way that oxygen on the top, and the other direction it's on the bottom. So they're going in opposite directions. That's what anti-parallel means. Okay. So here, this is just showing a kind of a graphical representation. Here is showing actually what it would look like. Again, it's kind of twisted. It's a helix structure. So it's not just 2 straight strands. It twists around itself. And again, I'll let you look through this a little bit more. But basically, this shows the beginning of replication. I'll show you in a different slide, in a more detailed slide, exactly how that happens. So DNA synthesis, basically the 2 strands open up, and then each strand becomes a template for a new strand. So the DNA, again, is made up of 2 strands. If it wants to replicate, those 2 strands separate, then each of those strands becomes 2 strands. Okay? And I'll show another graph on that too. So again, DNA synthesis, so this would be when a chromosome needs to replicate. right before a cell divides, it needs to make its one chromosome into two chromosomes because there'll be two daughter cells. So it's copied by an enzyme called DNA polymerase, and that's a very important enzyme. It's copied, again, DNA has two directions because it's anti-parallel. It can only be copied in one direction, 5' to 3'. I'll show you what that means in a second. It's initiated by an RNA primer. Basically, that's a little piece of RNA that is needed in order for the DNA polymerase to dock onto the DNA. So there's two types of synthesis. There's leading strand synthesis and lagging strand synthesis, and I'll show you what that means in the next slide. I'll talk about Okazaki fragments. Let's skip over that. All right, so here's DNA synthesis. Okay, so here is a parental strand, so this is what you're starting with. Okay, your double helix DNA. And it has to be replicated into two pieces of DNA. So what happens is it basically opens up like a zipper. Okay? And when it opens up, some proteins bind to it to kind of keep it stabilized and prevent it from coming back together. Okay, and these enzymes, there's enzymes that kind of unwind it and stabilize it to keep it together. And at that point, DNA polymerase, which is the enzyme that does the copying, can take one of these strands and start adding bases to it in order to replicate it. Okay, and it's adding bases in a complementary fashion. So every time it sees an A, it adds a T. Every time it sees a C, it adds a G. And it basically just replicates what this strand was up here. Okay? So that's leading strand synthesis. That's just going, as this open up, polymerase is just kind of following, and as it unwinds, it just keeps going and keeps replicating in that direction. Okay? Now, because DNA polymerase can only work in one direction, it can't do that on the bottom strand because it's anti-parallel. So this DNA strand is actually in the other orientation. So DNA polymerase, because it can only work 5' to 3', can't go in this direction. So what it does here is, as it's open up, it binds here and starts to fill in the gap. And then as it opens up more, it binds here and starts to fill in the gap. So it basically fills in these little fragments and then goes back and fills in another fragment and goes back and fills in another fragment. And so that's what's called lagging strand synthesis. Okay? And that requires a few things. It requires an RNA primer, and it requires these Okazaki fragments. And I'll talk about that later. But again, do you guys see the difference between leading strand and lagging strand? So leading strand is just continuous replication. It basically just binds once at the very end. It can continue all the way down, and as the DNA opens, it just continues. Whereas lagging strand, it has to keep binding as it opens and filling in the gap, and then as it opens more, it comes back and binds more and fills in that gap. Okay, so that would be lagging strand synthesis on that side. Okay, so bacterial DNA, again, is circular. So the way it replicates is there's an origin of replication, so that's where it first starts to split. Okay, so that double helix first starts to split at the origin of replication, and basically it just opens up like this, okay, and just starts to replicate in both directions down the side. So it starts here, starts to replicate like this. So you can see here, it starts here, and it just keeps going and opens up until it gets to the very end where it has a termination of replication, and then the two DNA strands split. Okay, so here you have two identical DNA strands that are identical to the initial strand. Okay, so again, the important thing here is that it happens in both directions. Okay. So let me show you a video to show you the overview here.